This is great. So I could give with the short time, I'll give summaries of our experience on pharmacokinetic and bioanalysis for peptide drugs. Okay, so because bioanalysis pharmacokinetic for peptide drug is some kind of challenge. So we meet some challenge. I'm gonna give some suggestion advice for your future study on the pharmacokinetic of peptide drugs. So later I'm gonna give a little bit about medicinal platform. Maybe could help those professors who have drug candidate to do co-development of peptide drug in China. Okay, I'm in Shanghai now, and unfortunately I cannot go to Singapore, or else I would like to go there. So very quickly, a little bit information about peptide drug and the like characteristics. Okay, as you can see from this slide, so many peptide drug in clinical use. So this is a great direction. I think today, so many professors here, this is a good direction. So a lot of pharmaceutical company need peptide drug. Okay, so but in order to understand the peptide drug, we need to know drug-like properties. Okay, and peptide drug actually belong to biologicals. Okay, compared with the chemical drug, a little bit of difference in the structure, especially on drug metabolism and pharmacokinetics. Okay, because peptide usually poor oral bioavailability or poor stability, but they are not metabolized by P450, and the tissue distribution really vary. Okay. I think it's kind of challenging to study pharmacokinetics of biological, including peptide drug. I listed 9 challenges here, including because the metabolism and the elimination scheme a little bit different from chemical drugs, and currently have not come up with some system for that. And also it's a peptide, so maybe formation of antibody, maybe have some binding protein, and maybe have a basic concentration in blood. Okay, so all those is further discovery, especially pharmacokinetic study for peptide. And today I'm gonna focus on a bioanalytical work on this. Okay, because as I mentioned, it's not a chemical drug, it's a biological, so it will be challenging to do bioanalysis for this. So bioanalysis for peptide drug usually for PK, TK, and also determine ADA, and also in a pharmacodynamic study. And 2 techniques are basically used. One, either equipment LC mass or ligand binding agent. I think most assay in our lab is related to LBA. Okay, it's because peptide, if over 3,000, it's going to be difficult to do in LC mass. Okay, and I think we met a challenge. I listed several challenges for your information. Firstly, is because peptide drug molecule either too small, it's going to be difficult to produce the antibody. Without antibody, it's going to be difficult to do LBA. Okay, so that's why we need to doing very well. Later, I'm going to be let you know how to overcome this challenge. Okay, and a small molecule weight and limited epitope number, so maybe affect the antibody production. Okay, so and this is interesting. If the immunogenicity is weaker and antibody is not easy to be prepared. Okay, if immunogenicity is strong and it's good to produce antibody, but highly positive instead of ADA in the animal body or a human body. So the second challenge is route administration, because most of the time peptide drug either sub-Q or oral. Okay, and usually low dose. So low dose causes low drug concentration in the blood sample. Okay, so they're gonna be need a very sensitive, bioanalysis method. Okay. And the third challenge is the effective dose. As I mentioned, the dose are going to be very lower than usually compared with the chemical drug, especially those of hormone or hormone-like peptide drug. Okay, just last talk, just a few milligrams per kilogram. Okay, and then also leading to limited number of drug concentration in the blood. The fourth challenge is a potential endogenous counterpart in the body. Okay, so one polypeptide molecule, because they are small and maybe they create an endogenous or exogenous molecule. When you have using assay kit, maybe maybe interference together. Okay, this is another challenge for that. And the peptide stability is also important. So one, not only to do bioanalytical work, but also when you prepare drug for PK, PD, or in vitro biological assay. Okay, so it need to be careful because it's going to be easy degradation and maybe thermal instability. And when you shake it, may be unstable. So those are physical instability, okay, including freezing instability. So when you study, prepare the drug and prepare if long-term, if thaw and melt, going to be very instability. So you be careful, okay? So if you cannot come up the repeated result, so thinking about physical instability. Now also have a chemical instability, including the amination, maybe oxidize, hydrolyzed, all those ones are chemical instability. So we need when you're doing prepared solution, also really be careful. Of course, in vivo also show those kind of instability. Okay. And also number 6 challenge is immunogenicity method. As I mentioned before, if highly immunogenicity maybe produce more antibody in the body, and then maybe produce you need an assay on ADA. Okay, so this is also important. So molecule labeling of drugs is very difficult because to set up the bioanalytical method. Now, how to, if we know those issue, okay, during bioanalytical work, so we have a strategy to do like that. For example, we're going to produce antibody. We're using different approach for immunization for antibody production. So there are several different ways in these slides. Okay, people, you're going to do in this, you can try. Okay, different way. The key point is, peptide antigen for immunization should be designed and prepared as soon as possible. Without the bioanalytical method, you cannot do in PK, you cannot do in toxicokinetic. So this is important. So homologous between species can be compared by using RAS tool. Okay, this is the first strategy. And second strategy is going to be eliminating endogenous interference. I know kids are going to be different, sometimes a little bit cannot differentiate very well. So endogenous interference is important. And this phenomenon also existing when antigen stimulate in the body. Okay, so you be careful on endogenous interference. The third strategy is going to be dealing with stability. When we know physical instability and chemical stability, maybe enzymatic stability. So if we understand this, so we can using different way to protect the stability. Or for example, we can using heparin anticoagulant, to do some increase the stability. Okay, and maybe going to be using EDTA or other things to inhibit the activity of other enzyme. Okay, so if we know the instability, we can solve the issue. And further, like a pH and maybe other ingredient also affect the instability. So when we know this one, we can easily prevent. If a pH is so we can prevent those susceptible and also heat acid related. Okay, so we can also overcome aggregation and absorption, all those kind of stuff. So unstable drug molecules should be prepared fresh and using immediately. So either daily basis or couple weeks. If you know the instability, you be careful. I know sometimes for research, we prepare maybe using 2 weeks or 3 weeks. That's going to be a challenge because you don't know the instability. So be careful on even for in vitro study. So, and for method, as I mentioned, okay, it can be need a value sensitivity. Okay, so this is based on analyzer platform, and a different way can do different LBA assay. So you can see try different way. Okay, and so also important need based on molecular labeling. Okay, different way to increase the sensitivity. Okay, so strategy based on sensor format design too. So assay format is also important. You can try the sequence or add reagents or also those one to improve the assay sensitivity. So there are many ways to improve sensitivity or analytical method. Okay, so you can based on a signal amplification system. You can using substrate change. Okay, so it's important. So high quality key reagent is also basic. Okay, and the technical experience is important. I know highly variation you working on LBA compared to LC mass. And as I mentioned, most of the peptide also need pay attention to ADA assay because when you get it into the body, you have produced ADA for submission to US FDA or Chinese NMPA. Also need the ADA assay. Okay, ADA also again is based on the molecular labeling. This is important challenge in analyzing the peptide immunogenicity. Okay, so this is important to work on ADA assay. Again, we can change assay format to make sure determination of ADA is value sensitive. Okay, so this also can be adjusted. So as we know that challenge, and we can have a strategy to solve those issues to make bioanalytical work value sensitive, reproducible, and this is going to be work under maybe GLP for toxicokinetic or non-GLP for PK study. Okay, so Medicinal have done many years on those kind of work. So I just quickly overview. So for you and hope in the future we can initial cooperation. I know professor usually identify one drug candidate, and if we're going to move forward to IND filing, it's not easy. Okay, so Medicinal are going to give a little bit of information. We have an 18-year experience. We now over 3,000 scientists here, and with near 1 million square feet lab in Shanghai. It's going to be in the future going to be a 2 million square feet lab. As you can see from this slide, we are building up 3 branches: drug discovery branch, and then you will identify drug candidate. We can do IND filing for like a drug development, basically CMC work for the production of peptide drug API and formulation, and further undergo preclinical research, including element study, DMPK study, efficacy, and GLP tox, and combined with the CMC and the preclinical research, then we can submit the IND. Okay, so then one got approved, can further get into clinical trial. So I hope next time when COVID-19 are gone, and I wish so many professors can come into Shanghai visit us. We have, as I mentioned, we have over 3,000 scientists here. We now provide service to over 1,400 clients around the world. as I mentioned here, we are glad. Within 5 years, we help our sponsors submit over 200 IND got approved international. As you can see from here, near 200 got approved in China, and you know, more than 20 drugs got approved in, you know, in other countries like US, Australia, and also EMA. Okay, so I hope those today there are several professors have a preclinical candidate. We can help you. We can do co-development, and then can license to the pharmaceutical company. So just last year, you know, we have 100 IND got approved. Okay, 85 in China, 14 in US, and in which either of IND is dual filing to China and the US FDA, and one in Australia too. So it's a great opportunity to make a transition from the discovery to the IND filing. Okay, so finally, I have 2 slides that actually promote our this team cooperation. We know have several professors have good drug candidate, right? Even I saw one pro drug or other like already got a clinical trial or approval drug. You can come into China. So we propose one we call IP, a CRO and a Chinese pharma working together. Okay, so we are reliable bridge and bond between foreign IP and a Chinese pharma, because both sides need the quality work at a CRO. Okay, so that's our proposal. I think which make me very happy today, because I saw so many drug candidates around. So we can help you to move on to the IND approval. So we actually have a several good case. Okay, you can check our you know, just a website. You can see there are 2 drug candidates actually IP generated in foreign country, and using medicinal IND filing capability and identify Chinese pharma to work together. Okay, so I know professors have lacked some funding, okay, but pharma have some funding, and medicinal can help get the IND done. Okay, so this is a very good case. Okay, I know today the main team at Singapore. Okay, I visited Singapore several times. So if I saw there was a good candidate, we can help you to bring to China and working together get IND approval. Okay, so can get a funding too, and sometimes Chinese company also promote a cooperation. So we have another drug candidate. It's called X8042. Actually, one of our friend, you know, licensing from Sweden. Okay, so whole IND package is done at the medicinal. Okay, now sold to Shanghai Pharma with probably, you know, 600 million RMB. It's great. So this model is great. I would like to promote working with the professor. Okay, and you know, medicinal majority our business came from biotech and pharma. But I'm really happy to try to work with the professor and move drug candidate, get into clinical trial, you know, through IND filing. Okay, so quickly summarize it, you know, so I think a peptide drug is right now one of the most important therapeutic medicine. Okay, so it's one way, and besides the peptide drug alone using as a single drug treatment, you know, patient, and right now very popular is, you know, PDC, right? I mean peptide drug conjugated, okay, antibody drug conjugate or peptide drug conjugate. This is one important direction. We do a lot of work on this. I know to do peptide drug, you know, I think a very challenging for bioanalytical work along with the pharmacokinetic work, because as I mentioned, not only difficult to set up a method either using LBA or, you know, LC method, but there are other challenges, instability, maybe endogenous interference, okay, maybe ADA. Okay, so very challenging on this. Okay, I'm glad that today with Lian Sang help connection with the professor in this team. So we We can provide an integrated service from drug discovery to IND filing. And again, we can have a different model. You know, at the beginning, we just provide either FTE-based fee-for service. I know professors have a lot of students to work on that, but you have a challenge to work on IND enabling package. So Medicinal have capability to do that. So I would like everybody remember Medicinal. We definitely can help you get funding from Chinese government, and also majority case going to be came from Chinese pharmaceutical industry. Okay, we have many cases, successful case, and thank you so much for giving me the time to promote Medicinal. So hope in the future we can have more cooperation. Thank you so much. Looking forward to see you in Shanghai.